In this video, we will be going through the steps in doing an In-Cell Western analysis. So in the project, you're going to go ahead and click on Create New Experiment, and you will choose Multi-Well Plate. And then we choose In-Cell Western Analysis. Now from here, we can go through the assay development steps, but for this video, we'll be doing what is termed target analysis. And so since the image has already been imported into the image gallery, go ahead and click on that, and then scroll down to the image that you need and select. And then start. So we're going to call this In-Cell Western Analysis. And then you can give it whatever description that is appropriate for this experiment. Now, we can either select a template that is in the template library, or we can create a new one. And if we create a new one right here, if you have seen the other video for the template library, I discussed that you can either create one here or in the template library for have it used for later. But I'm going to go ahead and use one of the presets that is already in here. And I'm going to click this one, this template right here, and click Select. And you will see here that we have background and then 8 different treatments for this Western. Now this is appropriate for the image that we have, but like I said, if you need to create a new one based off of a new experiment, just go ahead and click Create New and walk through those steps. Just briefly to show you how that is done, I'm going to choose the target analysis. I'm going to call this Test 1. In-Cell Western in a 96 well format. And you start off by setting your background wells. So if you have, those are your background wells. You have your positive controls. And then you have to give it a name. Click Next, select your negative controls. Click Next, and then if you have replicates, you can set these as different samples or different treatments. So I'm going to say treatment 0. Treatment 1. Treatment 2, and so forth as you go along. So you can set that up, and then this will be saved as a template in the template library. So we're just going to back up here. And we'll go back to the original experiment since we don't need to set up the template at this point. So I'm going to go back and select the template that we had chosen before. And so what's important to know with this is that the background wells are treated with secondary antibody only. So they don't have any primary antibody or any cell stain with it. So for this, if we need to, we can bring up the well so it's a little bit easier to see. Now, one important thing to note about this experiment. In this experiment, we have used the cell tag 520, and this was imaged on the Odyssey M. The process will be very similar for the CLX. You'll have the cell tag in the 700 channel and your target in the 800. But for this particular one, I'm going to choose the cell tag 520. If you are using the CLX, you would just choose the cell tag for the 700. But with this, we're able to get the normalization and 2 targets out of 1 image. Now, if need be, we can rotate the grid a little bit. We can increase or decrease the well sizes as needed. And we can also use trim value calculation. So if you have, if you by chance have any saturations in any of the wells, you can use the trim value to accommodate for that. And so now we're going to move on to the data analysis by clicking the next button. So I'm going to start a new chart on here, and I'm going to look at the fold change. And this is going to be for our phospho ERK on this image. And that is in the 800 channel. Click Start. And so the 0 treatment is going to be our control. And so that has been treated with no target drug. And then we have increasing amounts of drug target on here. These are twofold dilutions, starting off at 200. And then you have to name this series, and they should all be the same. And so I'm going to just simply copy and paste this to all of the series titles. And then you can view this chart as either a scatter plot, a line chart, or also a bar chart. And so they will give you the standard deviations since these are all groups together. We can also start another chart if we wanted to view the total ERK normalized against the cell stain. But for now, just for simplicity, we'll just go with this. Click on the next button. And then you get to the review and report. So this is the white light channel, and it shows you all the signal, background, normalized signal, and then all of the well information. And then it also goes through everything for the cell tag 520. And on any of these, if you want to export certain sets of data, you can click on this export button. You also have the option, in the quantify wells, you can look at the analysis table and pull up all of the data and then export these all together at one time. So all of these charts will have the images, all of the data tables, and if you do want to export the images by themselves, again, you just click on the export image button, and then you have options as far as TIFFs and JPEGs and resolution and so forth. Then down at the bottom of this report, it does have the charts for the phospho ERK that we generated, and then a composite picture as well. You can add your experimental observations, next steps, and summaries. If you want to export this as a PDF file, that PDF will contain all of the information that is on this report right here, just in one document. You can also export them as an experiment file, which will allow you to take the analysis to another computer for further review. Thank you.